Welcome to the debate. Right now, microbiome science is torn between two very expensive maps.
Very expensive maps.
Yeah. Imagine you're a cartographer in the 15th century. You have these broad, sweeping outlines of continents, and you desperately want to zoom in, read the individual street signs. But you simply cannot afford to send surveyors to every single alleyway on Earth.
Right, you have to choose your scale.
Exactly. And that brings us to the central controversy of modern microbiome science based on today's source material. Should research move beyond low-resolution marker gene surveys and transition toward high-resolution whole genome metagenomics as the new standard?
Or to put it simply, is depth worth the cost?
That is the exact question. Today, we're exploring the divide between the need for standardized, cost-effective ecological scale and the necessity of strain-level, mechanistic precision. I will be representing the position that 16S amplicon sequencing remains the essential, scalable backbone of microbiome research.
And I will be arguing the opposing view that whole genome shotgun metagenomics, or WGS, is the necessary future, and that 16S is essentially a legacy method of skewering critical biological realities.
We are definitely at an inflection point. For a long time, we've accepted these approximations of the microbiome.
Now we actually have the technology to see the machinery. The question is whether we're willing to completely change our approach to use it.
Let's lay the groundwork with how 16S actually operates, because the biochemistry behind it is exactly why it remains so powerful today. 16S sequencing relies on targeted PCR amplification.
Right.
We are specifically looking for the hypervariable regions, usually the B4 to V5 regions, of the 16S ribosomal RNA gene, which is found in bacteria and archaea.
It's essentially a barcode. You aren't reading the whole book, You're just scanning the barcode on the back cover to figure out what species you're looking at.
Exactly. We extract the total DNA, and we use primers. These are little starting blocks for DNA replication that are specifically designed to latch onto those bacterial barcodes. And the core biochemical advantage here is profound.
The host depletion.
Yes, those primers bypass eukaryotic host DNA entirely. If you're profiling a clinical specimen with extremely low microbial biomass, like a skin swab, or a biopsy from mucosal tissue in the gut, human host DNA can make up 90 to 99% of that sample.
Oh, easily. Sometimes more.
Right. But 16S completely ignores the human DNA. It enriches only the microbial signal. It acts as a massive biological filter.
A targeted filter, sure.
A perfectly targeted filter. And because of this, 16S enables massive scale. It becomes mathematically and financially viable to run tens of thousands of samples.
For epidemiology, yeah.
Exactly. In large-scale epidemiology, if you're trying to find a subtle signal across a vast human population, you need enormous cohorts to get true statistical power. That is how you make population-level ecological inferences.
I hear you. And while 16S was a phenomenal stepping stone to get us thinking about the microbiome, that biochemical mechanism you just described is exactly its fatal flaw.
How so?
Well, by relying on targeted PCR of a single marker gene, you're throwing away the vast majority of the biological data. Whole genome shotgun metagenomics doesn't use targeted primers. We aren't looking for a barcode.
You're just grabbing everything.
Exactly. We extract the total genomic DNA, randomly fragment it into small pieces, usually through mechanical shearing or by literally throwing an enzyme at the DNA to chop it up, and paste on sequencing tags. And then we sequence all of it indiscriminately.
Which means you are sequencing every domain of life.
Every single one. It's a multi-kingdom approach. You capture bacteria, archaea, fungi, and DNA viruses in one single run. But more importantly, 16S collapses critical biology because it fails to resolve the strain level.
You mean distinguishing between closely related bacteria?
Yes. Under a 16S marker gene survey, two strains of E. coli might look taxonomically identical. Their barcodes match perfectly. But biologically, for the patient sitting in the hospital bed,
The clinical outcome could be vastly different.
exactly. One might be a perfectly harmless, immune-modulating commensal bacteria just sitting in the gut. The other could be a virulent toxin-producing pathogen that is actively making them sick.
So you're talking about the functional differences between strains.
True clinical translation requires us to track mobile genetic elements, plasmids, and antimicrobial resistance genes. If a bacterium picks up a plasmid that makes it resistant to antibiotics, its 16S barcode doesn't change at all.
It still just looks like the same E. coli.
Right. Those resistance elements are driving the disease, and they are completely invisible to 16S.
Okay, the clinical importance of tracking a resistance plasmid is undeniable, I'll give you that. But we cannot ignore the reality of study design and the economics of sequencing.
Economics always dictate the science, unfortunately.
They do. We can talk about strain-level precision all day, but microbiome sequencing costs are dictated by library preparation complexity, reagent volumes, and reading depth. Deep shotgun sequencing requires tens of millions of reads per sample just to get enough coverage.
Because you're assembling the whole genome from tiny fragmented reads, it takes depth.
Precisely. And the economics of paying for tens of millions of reads dictate your cohort size. A large, adequately powered 16S study will always outperform a small, underpowered WGS study when we're looking for clinical associations in human populations.
But is it actually outperforming if the data is shallow?
Think of it this way. 16S is like a wide-angle satellite census. It captures the macroscopic ecological shifts of the entire forest perfectly. WGS is a street-level camera.
A very expensive street-level camera.
Right. It gives you incredible mechanistic detail on a single leaf, but it's far too expensive to deploy across the entire globe. If a researcher is looking for an odds ratio of 1.2 in a clinical association, a subtle shift in the microbiome, calculations show you need massive cohort sizes to detect those low-abundance taxa.
And you're saying you simply cannot afford to do that with deep WGS.
You can't. The math doesn't work.
Well, that economic argument holds up if we are strictly talking about deep shotgun sequencing. But you're ignoring the emergence of shallow whole metagenome shotgun sequencing, SSMS.
The shallow approach.
Yeah. SSMS completely disrupts that economic binary. We don't actually need 30 million reads per sample to get a robust profile anymore.
But you're intentionally under-sequencing the sample. You're sacrificing the very depth that makes shotgun valuable.
I'm talking about optimizing it, not sacrificing. By modifying the library preparation protocol, reducing the reagent volumes, and heavily multiplexing the samples on high-throughput platforms, we can target just 2 to 5 million reads per sample.
Okay, but 2 million reads is a fraction of what you'd normally want.
It is, but it brings the cost down to roughly the exact same level as 16S amplicon sequencing. And to use your analogy, shallow shotgun isn't the street-level camera. It's a drone hovering just above the trees.
A drone, okay.
You still get the wide-angle view, but with significantly better resolution than a satellite. Validation studies prove that shallow shotgun accurately preserves beta diversity matrices, species composition, and functional pathway profiles.
So you're claiming you get the census without the PCR bias.
Exactly. You get the census, but you're capturing the whole genome and totally avoiding the chimeras that inherently plague 16S amplification.
The drone analogy is great in a perfectly clear sky, but it completely ignores the biochemical reality of complex, messy environments.
Messy in what way?
Let me ask you what happens to that shallow shotgun drone when you fly it over a dense canopy where you can't even see the ground.
You mean high-host DNA environments.
Exactly. How can you justify shallow shotgun sequencing in low biomass environments like skin swabs or gut tissue biopsies?
It's definitely more challenging there.
Challenging is an understatement. If 95% of your sample is human DNA and you've capped your sequencing at just 2 million reads to save money, 1.9 million of those reads are going to be human.
You're left with a very small microbial fraction.
You're left with almost no microbial reads to actually analyze. Shallow shotgun completely fails in those scenarios without incredibly aggressive and expensive upstream host depletion techniques. And those techniques often destroy the bacterial cells you're trying to measure in the process.
Okay, that high host microbial DNA ratio is definitely the Achilles heel of shotgun metagenomics without physical depletion. I will readily concede that.
Thank you.
For a skin swab, where the bacterial biomass is tiny compared to the human cells, 16S has a massive signal-to-noise advantage. But let's look at what happens when 16S actually captures that signal. You hit a rigid taxonomic ceiling.
A ceiling dictated by the length of the read, sure.
Right. Standard 16S workflows only sequence a short segment of the gene, usually just 250 to 300 base pairs of that V4 region.
Which is enough for a barcode.
But they simply cannot capture the complete genomic variation required to differentiate closely related species. And the bioinformatics pipelines struggle to fix it.
Even with advanced algorithms?
Even when you run advanced error correction algorithms like Data2, which essentially tries to correct PCR errors to generate pure amplicon sequence variants, you still suffer from high false positive species assignments.
Because the sequence conservation is too high.
Exactly. The sequence conservation across those hypervariable regions is just too high. You are fundamentally capped at the genus or family level for many organisms. You might know a Bacteroides is present, but you have no idea which one.
I mean, 16S does rely on a shorter read, which inherently limits absolute resolution. I won't deny that. But both 16S and Shotgun Metagenomics share a massive vulnerability that dictates what we can actually see.
The databases.
Yes, they both rely entirely on the completeness of their reference databases to identify microbes. And this is a major underappreciated caveat for your preferred method. We really need to talk about the Shotgun database blind spot.
The blind spot being uncharacterized microbes?
Uncharacterized by WGS, yes. The paradox of the zymobiomic spike-in control is the perfect illustration of this. So researchers take Imteshala halotolerans, an environmental microbe that is completely alien to the human microbiome, and they physically spike it into a human fecal sample.
Okay, a known control.
Right. But when they run that spiked sample through standard WGS bioinformatic pipelines like Kraken-2, the algorithm misses it completely.
Well, because Kraken-2 relies on exact k-mer matching against its specific database.
Exactly. Whole genome databases like RefSeq or the Unified Human Gastrointestinal Genome Database are heavily biased toward cultivable human pathogens and biotechnology strains.
They represent what we've bothered to sequence.
Right. If there is no close genomic representative in the database, the algorithm essentially returns a blank stare. The shotgun reads go entirely unclassified. They are just wasted data.
And 16S catches it?
Yes. When you take that exact same spiked sample and analyze it using 16S, it successfully identifies it.
Because the 16S database is just older and broader.
Much broader. 16S databases like Silva and Green Genes represent decades of broad environmental curation. We've been using 16S to survey oceans and soils for 30 years.
It's a huge catalog.
And here's the crucial part. Even if 16S misses an exact species match, phylogenetic placement allows the algorithm to gracefully demote it to a higher taxonomic rank.
So it defaults to the family level.
Right. It might say, I don't know the species, but I know it belongs to this family. You don't lose the ecological data. With Shotgun, if the genome isn't in the database, the data is essentially thrown out.
Okay, that is a real issue for an uncharacterized Amazonian soil sample or a deep-sea thermal vent. If we're exploring an unknown environment, the Shotgun database blind spot is totally valid.
It is.
But let's ground this in clinical translation. The vast majority of the time, we're talking about human disease. Take colorectal cancer, for instance.
A heavily studied microbiome environment.
Right.
When we evaluate machine learning classifiers for detecting colorectal cancer, both methods can find the highly abundant oncogenic pathogen, Parvomonas micra.
Because it's a known, heavily sequenced pathogen. It's in every database.
True. But WGS provides a significantly richer dataset around it. When you compare 16S and WGS directly on human stool, the 16S datasets are structurally sparser.
In what way?
They routinely exhibit lower overall alpha diversity, meaning they report fewer unique organisms. By fragmenting the whole genome, we aren't just looking at a taxonomic name tag.
You're capturing the functional potential.
Yes, we can see the actual genes that Parvomonas micra is using to drive inflammation in the gut.
But if functional potential is what you're after, you don't actually need shotgun sequencing to get it. We have highly mature, predictive bioinformatic pipelines for 16S that bridge that exact gap.
Oh, you're talking about PICRAS-2.
Yes, PICRAS-2. It's an incredibly clever workaround. It places the 16S short reads into a pre-existing reference evolutionary tree. Then it performs hidden state prediction.
It infers the genes.
Essentially, yes. It looks at the closest evolutionary relatives and infers what genes should be present in your specific sample. It allows us to predict the functional metabolic activity of the entire microbiome using only the 16S barcode,
A prediction, not a measurement.
but a highly accurate one. Early validation studies showed incredible Spearman correlation coefficients above 0.8 between the functional predictions of PICRAS-2 and direct, expensive shotgun metagenomics sequencing. We can infer the functional pathways without paying for the shotgun depth.
I am so glad you brought up PICRAS-2, because that 0.8 correlation is one of the greatest statistical illusions in modern microbiomics.
An illusion. The math behind that correlation is rigorously documented.
The math is fundamentally flawed by permutation mathematics. Let's break down exactly why that high Spearman correlation happens.
Okay, explain the flaw.
Well, functional metabolic profiles naturally exhibit a much lower variance across biological samples than taxonomic profiles do.
Meaning the genes are more uniform than the species?
Exactly. Think about it. Every single bacterium has to replicate DNA. It has to synthesize proteins. It has to run core carbon metabolism to process sugar.
Basic survival mechanics.
Right. These are highly conserved housekeeping pathways. They are everywhere. Predicting that a microbiome sample has sugar metabolism genes is like predicting that a major city has restaurants. It's a baseline assumption, not a brilliant insight.
But the 0.8 correlation implies the algorithm is successfully tracking the nuanced variations between different samples.
That's what researchers thought until they rigorously evaluated it with a brilliant test. They took the dataset and they randomly shuffled the gene abundances across the biological samples.
They deliberately broke the biological association?
Yes, they deliberately broke any true biological association in the data. They assigned patient A's genes to patient B's 16S profile. Let me ask you what should happen to that 0.8 Spearman correlation when you shuffle the data and completely break the associations.
Well, the correlation should completely tank. The predictive power should disappear.
It barely moved. It stayed almost completely unchanged.
Wait, really? Because the baseline housekeeping genes are so overwhelming that they prop up the statistical correlation?
Exactly. The high correlation coefficient is a statistical artifact driven by the low baseline variance of functional profiles, not the actual predictive power of the algorithm to find nuanced metabolic changes. It's a mathematical mirage.
That is a harsh assessment.
And beyond the math, think about the biological mechanism. Algorithms like PICRAS-2 map against static reference genomes. They are utterly blind to horizontal gene transfer.
Because they can only predict what's in the reference tree.
Right. They cannot see a plasmid-board antibiotic resistance gene that just moved from one species to another under selective pressure in a hospital ward yesterday. If you want to understand how a microbiome is functionally responding to a localized selective pressure in real time, predictive 16S pipelines are flying totally blind. You are guessing, you aren't measuring.
Okay, the permutation math on PICRAS-2 is a powerful critique, and I will concede that relying on static genomes severely limits the ability to track real-time horizontal gene transfer.
It's a fundamental limitation.
But 16S as a field is not statically frozen in the short-read era either. The field is rapidly pivoting to third-generation long-read sequencing, which serves as a massive rescue for the taxonomic limitations you mentioned earlier.
You mean moving away from just the short V4 region?
Right. We are now doing full-length 16S sequencing using platforms like PacBio circular consensus sequencing and Oxford nanopore technologies, specifically their newer R10.4.1 flow cells.
The long-read revolution.
Exactly. Instead of snipping a tiny 250 base pair barcode, we are capturing the entire 1,500 base pair 16S gene. That includes all 9 hypervariable regions, plus the conserved regions bridging them.
Which definitely gives you more data per read.
By doing this, we completely break through that genus-level ceiling. We are routinely achieving species-level and sometimes near strain-level resolution, all while maintaining the massive cost benefits and host DNA filtering of an amplicon approach.
The move to full-length 16S is definitely a massive upgrade over short-read amplicons. The accuracy on those new Oxford nanopore flow cells is impressive.
It really is.
Especially when paired with deep learning base callers, the algorithms translating the raw electrical signals into DNA letters, they are pushing past 99% raw accuracy.
But you can't ignore the platform-specific biases that introduce noise.
The error profiles are different between the technologies, yes.
Radically different. ONT-based callers filter out a significantly higher percentage of reads in complex regions due to sequencing errors compared to PacBio. But we can adjust for that computationally.
Still, in comparative studies looking at complex soil communities, PacBio workflows filtered out maybe 5% of the reads. The ONT workflows filtered out over 16%. That is a massive chunk of data lost just to error correction.
That is a known caveat of long-read technology, which simply means ONT runs require a slightly higher raw read input to achieve an equivalent post-filtering depth.
It's still lost data.
But we're compensating computationally. We have new specialized algorithms like EMU. Instead of traditional matching, EMU uses an expectation maximization algorithm.
Like a statistical autocorrect?
Essentially, yes. It acts like a highly sophisticated autocorrect that uses statistical context clues to fix messy long-read data. It specifically lowers the false positive rates for ONT long reads. But the broader point is the biology.
Which is?
We can achieve species-level resolution without abandoning the core benefits of amplicon sequencing. We still get selective amplification away from host DNA and vastly lower computational overhead compared to Shotgun.
But even with full-length 16S, even with flawless long reads and perfect error correction, you are still inherently restricted to a taxonomic profile.
You still think it's just a barcode?
You still only have a name tag. If the goal of the study is to map actual metabolic pathways, or to track novel gene discovery, or to find virulence factors, you are still hitting a wall. This is why hybrid assembly approaches in shotgun metagenomics are where the true frontier is right now.
Combining short and long reads?
Exactly. In highly diverse environments, short-read shotgun struggles to resolve repetitive genomic regions. It's like trying to put together a puzzle where all the pieces are blue sky.
Right. Short reads can't span the repeats.
And long-read shotgun has higher error rates that cause frame shifts in the genetic code. But when we combine them using the ultra-accurate short-read Illumina data to scaffold and polish the long-read Oxford nanopore assemblies, we get incredible results.
You're talking about MAGs.
Yes, we can recover metagenome-assembled genomes, or MAGs, with incredible contiguity. We are pulling high-quality, fully assembled, continuous bacterial genomes directly out of complex soil or gut samples without ever needing to isolate or culture them in a petri dish. That is true discovery.
And building high-quality metagenome-assembled genomes from a hybrid approach is undeniably a phenomenal feat of bioinformatics. I'm not taking that away from you.
It's the future.
But let's ground this in reality. It is astronomically expensive, and the computational power required to run hybrid assemblies on thousands of samples for an epidemiological cohort is crushing.
It's computationally heavy, yes.
It requires massive server farms and computational pipelines that most clinical labs simply don't have, which is really where this debate settles, I think. We need to step back and map the operational boundaries we've established today.
Because there isn't one universal tool for every biological question.
Exactly. 16S amplicon sequencing remains the undisputed choice for low biomass samples where the host-to-microbial DNA ratio is exceedingly high, like skin, lungs, or mucosal biopsies.
Due to the biochemical filter?
Where shotgun reads would simply go unmapped due to database blind spots. And fundamentally, it provides the sheer cost efficiency required to power massive epidemiological cohorts. For sheer statistical power, sure. If you need 10,000 patients to find a subtle clinical association, 16S is how you get the statistical power to find the truth.
And I would set the boundary by reinforcing that if you are asking a mechanistic biological question, whole genome shotgun metagenomics is mandatory.
For the actual machinery?
Right. If a patient is sick, predicting their microbiome's function based on evolutionary assumptions isn't enough. If you need to map strain-level virulence profiling, if you need to identify plasmid-borne antibiotic resistance, if you're tracking horizontal gene transfer, you must use WGS.
Or building clinical biomarkers.
Exactly. Building FDA-grade biomarkers based on actual metabolic reality rather than taxonomic approximation requires WGS.
The sheer complexity of the microbiome requires us to move beyond the wide-angle ecological senses and into the actual genetic machinery driving these systems.
It is a remarkable landscape to navigate. Ultimately, the best scientific insights don't come from forcing one platform onto every problem.
Never works.
No, they come from clearly defining the biological question and then aligning that question with the correct biochemical tool. And to make things even more complicated, we haven't even touched on the frontiers of RNA metatranscriptomics.
Oh, wow. Yeah, that's a whole other level.
Right, where we stop looking at the static DNA blueprint entirely and start measuring the active gene expression, which adds an entirely new dimension of chemical instability and complexity.
It certainly does. We're moving from a static map to a live traffic feed. The tools are evolving as fast as our questions, and the data is only getting richer.
Which brings us back to our 15th century cartographers. Whether you need a massive satellite view to understand the slow migration of a continental forest, or an incredibly precise street-level camera to read the specific warning signs in a single alleyway, the map of the microbial world is getting clearer every single day.
It is.
The question you have to ask yourself before designing your next study is simply this: what scale of truth are you willing to pay for?